EBSCOR, the experimental program to stimulate competitive research, is a National Science Foundation grant program. The goal of EBSCOR is to strengthen science and engineering research and education throughout the United States, and to enable states like Wyoming to be nationally competitive in these areas. EBSCOR aims to improve scientific infrastructure in Wyoming and build science, technology, engineering, and math research capacity across the state. One way Wyoming EBSCOR helps strengthen science research and education is through student programs such as the Summer Research Apprentice Program, also known as SRAP. SRAP is an intensive 6-week paid research experience for high school sophomores, juniors, and seniors aiming to attend college. It offers students a hands-on research opportunity in science labs across the University of Wyoming campus. Students work one-on-one with a faculty mentor throughout their time in the program. SRAP not only provides students with a paid internship and real research experience, but also helps prepare them for college life.
Mr. Danny Tran from Arvada, Colorado, and he will be presenting the importance of plant stress.
Hello everyone, I am Danny Tran, and I'm doing the importance of plant stress. My mentor is Dr. Brent E. Ewers, and I am from the botany department. Plant stress may be detected by many different methods. This includes the detection of cavitation and water release in the soil. Cavitation can also be caused by certain types of elements. Cavitation occurs when the tension in the xylem column becomes high enough to pull air into the xylem, or when the column gets filled with air. The xylem is a tube made up of tissue cells that helps bring water from the roots and transport it up to the plant's canopies. When cavitation occurs, an air bubble gets trapped into the xylem and ruptures some of the xylem cells. This causes the plants to have a lack of water in their canopies and end up dying. A vulnerability curve is based off of cavitation. It is a plot depicting the decline in conductance with increasing temperature. A moisture release curve, also known as a soil moisture characteristic curve, depicts the moisture of the dirt to the evaporation. This is important because it can show us how much water is available for surrounding plants and how much water is given off for a certain type of soil. My objectives is to create a graph of vulnerability in plants due to cavitation, to create graphs on water release in the soil, and to explain how they're important in plant stress. The materials I used was a hydraulic system, also known as the high-resolution flow meter, soil samples, stem samples, a WP4, also known as the dew point potential meter, a balance scale, and a centrifuge. Here we have a mountain part of Wyoming. The little red dot represents Laramie, and these blue dots represents our glacial till sites. At the glacial till sites, that's where I have retrieved my soil samples. To get my soil samples, I use a soil core driller and drilled 0 to 15 centimeters into the earth and gather this sample. Afterwards, I gathered a sample that's 15 to 30 centimeters. This yellow tack here represents Chimney Park, and that's where I have retrieved my stem samples. To get the stem samples, a 20-gauge shotgun was used to shut down branches, and then we collected our samples. In the lab, to find our water release curves, we use the dew point potential meter. And what that does is it uses a method of psychometry called the chilled mirror technique. How this works is as you place the sample into the WP4, or dew point potential meter, the water evaporates into the space above the sample. There's a mirror inside the WP4 that cools, and that cooled mirror then condenses the water vapor. The results displayed on the screen are based on the difference of the soil temperature and the condensed water vapor temperature. After recording the temperature, time, PF, and megapascals, we waited a certain time interval, such as 30 to 45 minutes. We then repeat this process until the soil water potential has become stable or dry. Once the soil has become completely dry, we finally take in the final weight. Over here we have our trusty dew point potentiometer, and over here is a typical balance scale. For our vulnerability curves, we retrieved our samples from a certain type of gymnosperms called the lodgepoles. Once we got back to the lab, we recorded the stem's info, such as the name, date it was taken, length. Then we take the temperature, the height difference between the meter pipe and the scale pipe, and if there was any pressure applied, we would record the pressure. We then use the high resolution flow meter, and what that does, it allows water to flow through a certain sample. It's also called a hydraulic system because they also measure the flow before allowing water to flow through, during, and after. After we finish putting in the high resolution flow meter, we then place the stem into a centrifuge and apply pressure for 4 minutes. What a centrifuge does is it spins the sample at a certain RPM, rotations per minute, and creates tension. The reason we use a centrifuge is because during a drought, soil becomes so dry that it starts to absorb water, sometimes stealing water from the plant xylem. It also makes it harder for any water to leave its content. This creates tension on the bottom of the xylem because the water is being pulled out from the bottom, but the plant still wants the water to stay in and go up to the canopy. But at the same time, there's atmospheric drought, which is meaning the plant has to transpire and water is being given off from the canopies. So the xylem is being applied pressure on both ends. As the centrifuge spins, both ends of the xylems are applied pressure and tension. This is a high resolution flow meter or diagram, and all the water taps are positioned in direction of flow. The stem is placed so the canopy is on the right side, because this way, as we put in the water flow, it'd go in through what would be the roots, through the stem, and what would be the canopy, and into our scale. Here are my results for my flow characteristic curves, or water release curves, and my vulnerability curves. The reason why these are all connected is because they all use negative megapascals. What is negative megapascals? It's almost like the opposite of pressure. Instead of applying pressure, you're doing more of a sucking motion, or like a vacuum. This causes a tug of war between soil and plant to see who gets the water. And dry soil tend to make plants to use more tension. And if a plant uses too much tension, the xylem will cavitate. As you can see, that on the side you see percent gravimetric soil water content. What that is when you have a block of soil or a soil sample, not all of it is soil, but there's also water and air particles in it also. Underneath 5%, you can see at negative 2 megapascals. That is where most of the Engelmann spruce or our branch samples would cavitate because they have lost all their conductivity. Predictions can be made on which soil is fit for many crops. Vulnerability curves can determine which plant cavitates more often. This is important because then farmers may know which crop fits which soil. You would not use a crop that cannot take much tension and give it to a type of soil that does not give much water. This may also help with irrigation, because if the farmer knows how much water his soil has, then he doesn't need to water it as often, or maybe he needs to water it more. Plant stress also helps us tell which soil is more affected in drought. Like sandy soil evaporates much quicker, but as for clay soil, it retains water better, which is good because it keeps water during a drought, but since it retains water better, it forces a plant to use more tension on the xylem. If I have to do this project again in the future, I'd hopefully have a longer time span, because due to time restrictions, we were unable to take more data or samples. As we were taking soil samples, most of the times we were going deeper into the earth for 15 to 30 centimeters. A large rock or some root would be in the way, so we have to change location and spend more time finding a good spot. Sometimes the centrifuge was a problem, because due to the large amount of pressure, the stems might snap, and you will never know if you're really at the perfect rotations per minute for a megapascal. I'd like to acknowledge the Summer Research Apprentice Program, the NSF Wyoming EPSCoR, and the University of Wyoming. Dr. Brent Eulers, Tim Ashton, Lena, David Reed, and Scott Peckham. My fabulous lab partner, Martin Bauer. All the undergrads and grad students, like Heather, Willie, Ben, Caleb, and Rachel, and Sam. I would like to thank the staff and students of SRAP 2013, and last but not least, my parents. And my references? Questions? Yes?
If I understand you correctly, this type of research would assist people in agriculture for having better practices of how much to irrigate and which plants to plant where?
Which plants, which crops fit which type of soil.
How long between your collection of your logical time and your conductance? I'm sorry, what time elapsed between the time that you collected your sample and the time that you actually performed that experiment? And do you think that could have an impact on the data that you see there?
After we got our samples, my mentors put it in a refrigerator, and I'm not sure, like, if I'm not sure we're using like those exact samples, or maybe some samples that are collected earlier. But if it was earlier, then we did recorded the time and date that we got those samples. Yes.
So you mentioned the shop that did you get a ship that, or?
Sadly, no. Thank you.